Hi, welcome back. So, I think this is the last of the series of videos for the topic Introduction to Immunology. So, hopefully by now you have an idea of the functions of each of the immune cells. Okay? So, hopefully you can tag or your group mates, or you have an idea of which of these immune cells most likely resembles you. Okay. As for me, I'll just keep it to myself. Anyway, moving on. So, for this video, we will be talking about the immune response itself, okay, the features of the immune response, or rather the phases of the immune response. The objectives, we will discuss the phases of the immune response, the processes in each phases, and the regulation of the immune response. And the second part of this video will be about the clinical correlation. So we will briefly look into the different clinical conditions that would affect the immune system. Again, hopefully you can remember what happens when an antigen is presented, or when an antigen is taken up and presented by the antigen presenting cell. So it will be phagocytosed, and then the APC will display the antigen on its surface together with the MHC class 2, and then this will be recognized by the CD4 positive T cells, which are your helper T cells. The helper T cells now would undergo proliferation. So it will proliferate to become your memory T cells, and at the same time an effector cell. Now, being an effector cell, okay, it will activate the other immune cells directly or through the secretion of interleukin 2. Okay, so one of the cells that it can activate would be the B cells. So in this case, when your B cell is activated, it will again proliferate and differentiate to become your memory cells, memory B cells, and plasma cells. The plasma cells now would be the ones secreting the antibodies specific for that antigen presented by the APC. Now, this is a picture showing us the comparison, or how our immune system reacts when it initially encounters the antigen, and if it subsequently encounters the same antigen in the near future. So if it initially encounters the antigen, this is termed as the primary response. Okay. So same way as what I've shown you before. So the antigen can either be taken up by the B cell, or it can be taken up by the APC. The APC activates the helper T cell, which then activates the B cell. Now, the main point here is that once the B cell is activated, it will proliferate again to form the clones. The clonal differentiation can be a memory cell and the plasma cell. The plasma cells would produce the antibodies for that antigen. Okay. Well, not shown here would be the other processes involved, which example would be the class switching and the increase in the affinity of the antibodies produced. Okay. Now, just take note that these memory B cells, okay, this is a minor fraction of the clones coming from this set of lymphoblasts. Okay. But they are important because when these memory B cells, subsequently encounters the same antigen in the near future, which can be months or years later, take note of what happens. the memory B cells becomes activated, okay, and it will proliferate and differentiate into a set of clones again. Okay. Again, into some of it will become memory B cells again of the same type as this memory B cell. And if we look at the plasma cells, the number of plasma cells would be greater than the initial response. So if we try to graph the immune response, so the x-axis here would be the time, and then the y-axis would be the titer or the concentration of the antibodies in our blood. This would be the primary response. Okay. So after some time, the antibody levels goes down, but on subsequent reinfection during a secondary response, take note of what happens to the antibodies. It becomes greater. Another difference between the primary and the secondary response is that in the primary response, the antibody is composed, or the antibody is initially an IgM, and after a few days it will switch into the IgG here, as compared to the secondary response, wherein the characteristic of the antibodies produced here, or the type of the antibodies produced, would be the IgG specifically. These are the high affinity IgGs. Okay. So again, this is a table showing us the comparison of the primary and secondary response. The lag here means the time it takes. This is like latency in the nerve conduction velocity, or EMG. So after antigen exposure, there is a lag time before the antibody level increases, and this is usually 5 to 10 days during the primary response. But if you look at the secondary response, the lag day is usually less. It's 1 to 3 days lang. So it's safe to say that the secondary response is usually more rapid than the primary response. I've mentioned this one before. The peak primary would be smaller compared to the secondary response. And then the type of antibody is that IgM first, and then becomes IgG for the primary. And then for the secondary response, it is predominantly IgG. And in terms of antibody affinity, during the secondary response, these are the antibodies that are of higher affinities to the antigen. Okay, so this is a characteristic of the response, primary response. So this is where the antigen is introduced or exposed, or the body is exposed to the antigen. Okay, and then there will be a lag time. So at this point, the innate immunity works, and then the adaptive immunity starts to kick in. And then after several days, the antibody goes up, or the antibody level goes up. So there's a process known as your class switching, wherein the antibody type during the primary response changes from IgM. So from an IgM type, it becomes an IgG type. Okay. So usually this IgM would peak maybe 5 to 7 days after the exposure to the antigen. Okay. And then after that, it will eventually go down. For the IgG, it will start to peak in more or less on the 7th day. Yeah, it will start to rise, and it will eventually peak more likely 2 weeks after the exposure to the antigen. Okay, and then eventually it goes down after a month. This is your class switching. Okay, now if we look at the curve, okay, we can have several phases of the immune response. So you have the lag phase, and then you have the logarithmic phase, log phase, and then the plateau phase, and then the phase of decline. So there. Now the lag phase is the phase where no antibody is detected. Okay, again, this is from the time the antigen is introduced or the body is exposed to the antigen up to the point where the antibody level starts to rise. Log phase is the phase wherein from this point to this point, okay, it is characterized as a phase wherein the antibody logarithmically rises. Okay, so it will rise. Okay. And then the plateau phase is where the antibody level stabilizes. So there is a constant level of antibody. And then after that, there will be a decline phase wherein the antibody is being cleared from the body. Okay. Same picture, the phases of the adaptive immune response plus together with it the processes involved in each response or in each phase rather. So here. So usually at the lag phase, so it's where the innate immunity works. So there will be phagocytosis of the antigen, and thereby it will be presented to the helper T cell. So it will be in the recognition phase. So the recognition phase will fall under this log phase. During the log phase, this is where the clonal expansion and subsequent activation of the B cells would occur, such that the level of the antibody rises. During the plateau phase, this is where the level of the antibody remains the same or stabilizes, and usually this is where the antibody works, or the effector phase or effector response. And then after that, the declining phase is where the antibodies are being cleared from the body. Okay. Now, in terms of how our immune response is being regulated, so number one, it is influenced by the major histocompatibility complex. So you've seen before that for the T cells to be activated, the antigen must be presented together with the MHC. And aside from that, there should be a expression of the co-stimulatory proteins. Okay? There could also be release of activating factors from the innate immunity, which would activate the cells in the adaptive immunity. And there are regulatory immune cells. So of this, the most important would be the regulatory T cells. So this is the pathway by which the regulatory T cells work. Okay, so the regulatory T cells, these are a subset of helper T cells. So take note, they are also CD4 positive. So on the surface, there's also CD4 proteins, okay, and an additional CD25 protein. So this is the marker for regulatory T cells. Okay, take note that for the regulatory T cells would express this pathway, the FOXP3, which is an important pathway in the regulation of the immune system, because studies have shown that when they try to inhibit this FOXP3 pathway, what happens is that there will be overactivity of the immune cells. Either it becomes hypersensitive. This is in animal models. So there would be hypersensitivity reactions in mice or autoimmune diseases. We'll talk about these two conditions later on. Okay, so once this FOXP3 is activated, this pathway is activated, the regulatory T cells would secrete anti-inflammatory cytokines, which would inhibit the response of which would inhibit the helper T cells, okay, the cytotoxic T cells, and other cells like B cells and natural killer cells. However, for FOXP3 to be activated, it must require other factors. Example would be L-selectin and the anti-inflammatory hormone glucocorticoids or cortisol. Now, there are other factors that would contribute to the immune response or to the immune system. So, of course, diet. If you have malnutrition, you'll have a decrease in the synthesis of your proteins, which could affect the metabolism of your immune cells as well as the production of your immunoglobulins. For gender, they would say that the presence of the female sex hormone estrogen would have the following effects. And aging. Okay, so these are the consequences of the aging process. So generally, as a person ages, okay, the immune response would decline. Okay, but take note, you have the following increase. So more or less, this counteracts the decreasing levels of these factors in this person's immune system. Okay. Again, this is a reminder to stretch. So you can pause the video and again stretch and have a break. Okay, next part would be the clinical correlation. So we will look at this for clinical conditions affecting the immune system. First one is what we call as your hypersensitivity reactions. Now, in your hypersensitivity reactions, what happens here is that the immune system responds to a normally, take note, normally harmless substance. Example of this would be pollen, dust, certain drugs, etc. And act as if it was a threat. Okay. For this case, the one that induces the hypersensitivity reaction or allergic response is what we call as your allergen. Now, there are 4 types of hypersensitivity reactions. So the type 1, this is termed as immediate, meaning to say upon exposure to the allergen, you have the symptoms immediately. Okay. And this is mediated by IgE. Take note, this is your IgE-mediated hypersensitivity, wherein you have the allergen, which binds to the IgE, and then the IgE is then recognized by the mast cell through the Fc portion. And then upon binding to the mast cell degranulates, releasing histamine. So the examples of type 1 reactions would be could range from a simple allergic response, asthma, and to this condition known as your anaphylactic shock, which can be fatal. The other type of hypersensitivity is what we call as your cytotoxic type, okay, wherein the antibodies would react with the antigens in our own cell. Okay. In a cell, not necessarily our own cell. Example of this would be ABO incompatibility. So for example, the donor is, let's say, type AB, or let's say type B, okay, and then the recipient is type O. Okay, remember, the recipient is the one that would contain the antibodies in the blood. So if the recipient is type O, the antibodies present in that person's blood would be antibody A and antibody B. Now, if the donor is if the donor is type B, which contains, remember, the blood type corresponds to the antigen present on the surface of the red blood cell. So, if the donor is type B, it means to say that on the surface of the RBC, it has B antigen. So, when this B antigen interacts or is encountered by the antibody B, okay, the RBC would be destroyed. So there is cytotoxicity. Next type would be the type 3, which is the immune complex. So the immune complex here can be an antigen bound to the antibody. It can be this antigen antibody plus the complement system. Okay. So what happens here is that the complex are deposited in certain areas in the body, resulting now to a hypersensitivity reaction. Okay, example would be this one here. So this is the immune complex. It gets deposited to a certain wall. Let's say this one is vessel wall. Okay, and because of this, there could be attacks of other immune cells. Type 4 is what we call as your cell-mediated hypersensitivity. Okay. So this is usually mediated by, or this is being a cell-mediated, this is mediated by your cytotoxic T cell and your helper T cell. So what happens here, what happens here is that there is a delayed type of reaction because there is activation of the T cell. Example of this one would be the tuberculin reaction or TB, tuberculosis, contact dermatitis, okay, which are delayed reactions. So this delayed reaction can be more than weeks or days or weeks or months after the exposure to the antigen. For immunodeficiency, this can either be congenital or acquired. When we say congenital, there is a varied inborn error. So meaning to say, our body has, or this person's body has difficulty in producing certain immune factors. Example would be if it cannot produce the gamma globulin in the antibodies. So there will be decrease in the immune response. Okay? Or it can be acquired. When we say acquired, there are factors, drugs, radiation, clinical conditions that would interfere with the immune response. Okay? Of this, take note of cancer, okay? because there is failure of the immune surveillance, meaning to say the cancer cells tend to evade or can evade the surveillance done by the immune cells. They can hide from these immune cells. Another thing is that these HIV/AIDS, which primarily affects the CD4 positive cells. So if you may have noted, one of the diagnostic tests for HIV is, or not diagnostic test, but to check for the response to the treatment in HIV/AIDS is to look at the CD4 count, or the number of the CD4 cells. For immunodeficiency, please take note, since the body is not having a normal immune response, they are at risk of having recurrent or opportunistic infections. So, usually those that are not normally present, example would be fungal infection, okay, it can be present in immunodeficiency states. Organ transplants. Okay, so there are several types of organ transplantation. When we say autograft, it means to say that the same tissue is taken out and then transplanted to another area in the body. So same person. An isograft, when we say isograft, the graft or the organ would come from a genetically identical donor. Swerte if you have a twin, an identical twin. Okay. An allograft is from another person. For example, if you're talking about human being, another person, okay, not necessarily blood related. And when we say xenograft, it's like a game. Xenograft, the graft or the organ or the tissue would come from another species of an animal, or not from an animal. Okay. Example would be the valves, the porcine valves that are transplanted into the heart. Of this, since this can be viewed by our immune system as something foreign, well, except for the autograft, since it's from the same person, these are at risk to develop rejections. Okay. So this is usually mediated by the immune system, specifically the cytotoxic T cell. Now, the most compatible, or rather the way to determine the risk of rejection is, number one, we check for the blood type. Okay, they should have the same blood type, and then after that we check for the MHC. If the MHC are compatible, okay, most likely the tissue will not be rejected. Okay. So of the types of organ transplantation, the one that has the most compatibility in terms of MHC would be the autograft, followed by the isograft. And then the most incompatible, since the tissue would come from an animal, would be the xenograft. Okay. So these are the pathways by which the organs are being rejected. So there are three mechanisms. Number one is there is lysis of cells. If the antigens are being presented by the MHC or with the MHC1, so it will be targeted by the cytotoxic T cells. Another is that there could be antibodies produced against this foreign or tissue, let's just say tissue. And lastly, the innate immunity can also play a role, specifically the macrophages and other phagocytic cells. So take note, organ rejection or organ transplant rejection can be considered as a delayed type of hypersensitivity reaction, so type 4. Now, for autoimmune diseases, this is what happens here is that there is production of an immune response against our own tissues. So what happens here is that there is an inability to distinguish self from non-self. So most likely there's a problem in the education during the development of the T cells and B cells. Examples, there could be antibody to receptors. Examples would be the following: myasthenia gravis, Lambert-Eaton disease, and type 1, wherein you have autoantibodies to receptors. Another thing that can happen is there could be antibodies against cellular components. Example would be SLE. Right? Selena Gomez, right? She has systemic lupus, erythematosus. Okay. There could be also antibody against platelets resulting to ITP, idiopathic thrombocytopenic purpura, and there could be also antibodies against the thyroglobulin resulting to hypothyroidism. Okay, so in summary, what we have discussed would be the different phases of the immune response: the lag phase, log phase, plateau phase, and the decline phase. Now just remember what happens in each phases. Okay? We've also looked at the regulatory mechanisms for the immune response, specifically the regulatory T cell, or the importance of the regulatory T cell. And we've looked at the different clinical conditions that would affect the immune system. So hypersensitivity reactions, so you have 4 types. Okay. You also have immune deficiencies, autoimmune disorders, and organ transplantation, and rejection. Okay. References for this one, primarily I used, I also post the main references in Canvas. The main ones I used would be the basic immunology. For this one, please read chapter 1 and Guyton and Hall. For Guyton and Hall, please read chapter 35. If you have questions, you can post it in the discussion board I've published in Canvas also. Okay, so here the references. So before we end this, so I'll share this Facebook group. I just find this funny because you have a lot of memes about immunology. So majority of the memes you've seen in this video and in the previous videos would come from this group. Okay, so life lessons in physiology. So life with God is not immunity from difficulties. So it's not protection or defense against difficulties, but rather it is peace in difficulties. And on a lighter note, share ko lang to. So if you don't struggle, diba? Remember Magikarp, pag naubos na yung attack niya, struggle na lang. So if you don't struggle, you don't improve. You won't become Gyarados.